Yeah, so next part we talk about cancer epigenetic and epigenetic therapy. If you remember from Collins's mutation analysis and also the subtype analysis, people found that when we started doing high throughput sequencing on the tumors to look for mutations, one of the biggest surprise was they found a lot of mutations on the epigenetic regulators. So this is a review paper published in Cell where they listed, the rows are the different cancer types and the columns are the genes that are recurrently mutated in those cancer types. And you can see some of them are gain of function mutation, some of them are loss of function mutation, some are translocations, and some are copy number changes. You can see here, the copy number changes are very few. See the ones that with the little circle inside, they were really, really small numbers. So previously, if copy number is important, people would have found it from the microarray days. They can look at copy number directly from array, but they were not found before. It's only when people started doing sequencing to look at specific mutations, they realize, oh, these genes are mutated or translocated. And you can see there are a lot of these epigenetic regulators involved. And so depending on the different mutations, there are different situations. The first is methylation changes in cancer. So do you guys remember where DNA methylation happens in the genome? Sam? So DNA methylation happens a lot on the CPG island, and these are usually in front of genes. They control gene expression. If the promoter of a gene has CPG methylation on the CPG island, this gene will be off. And in cancer, very often we see hypomethylation. So if this gene didn't really need to turn on, they were usually suppressed. But somehow, kind of, you open the door in some sense. The promoter of these genes in normal cells are methylated, but somehow in cancer they become hypomethylated, and these genes become on now. For example, some embryonic stem cell are going through uncontrollable growth, right? So that's one. The other case, majority actually of the DNA methylation happen in the repetitive regions of the genome. Remember, a lot of these really highly repetitive regions are viruses that used to come in the genome and copy itself multiple times, and they jump around, right? So DNA methylation is a way to kind of silence them. They don't allow these genes to turn on. And so actually half of our genome are these repetitive sequences that are normally DNA methylated. And so these are really silent. Somehow in the cancers, it's like, oh, the cell can't really control its territories anymore. A lot of these massive regions become, sorry, I made a mistake. So, a lot of these regions that are traditionally in normal cells methylated become also hypomethylated, which basically a lot of regions become activated as well. So, sorry, the promoter actually can be hypermethylated. So these are genes that are potentially like suppressing cell growth, or check, make sure that the cells normally are with each cell cycle, they examine to make sure everything seems okay on the breaks. And so those genes very often are hypermethylated, meaning that you do not, the cancer cell don't allow those genes to be expressed anymore, right? So in some sense, the brakes are taken out. The widespread regions are more hypomethylated. These are the kind of the massive regions. They are kind of becoming out of control. And so, so actually TCGA did a DNA methylation. Initially, they used some array platform. These are much, much cheaper than the whole genome bisulfite sequencing. In the epigenetic module, we talk about sequencing using bisulfite sequencing, but for TCGA, they just use a lot of arrays. We don't need to do the animation here. So you can see in the early days, they were just mostly testing CPG island, but later in the later arrays, I think majority of the samples now have this new type, which has the CPG island, has the promoters. It covers a little bit of the gene body and also the promoters and also some intergenic regions as well. So in homework 5, you actually look at the promoter DNA methylation as well. But yeah, so all the types have this new 450 array profiled. Very limited number of TCGA samples have the bisulfite sequencing. And so in homework 6, we are also asked you to look into this situation as well. And so when people started looking at DNA methylations, we mentioned briefly that before, if you look at subtyping information, most people use gene expression or protein expression as subtyping. However, sometimes when people look at the DNA methylation patterns, you can also see some differences. So this is just to look at the promoter methylation of genes. So each row here is one gene, and the columns are different cancer types. And so you can see these regions are mostly methylated. These ones are mostly unmethylated, but you can see also the samples differ around, different blocks in here. Even for these genes, they are much more methylated in this first subtypes, right? And so this one is called CPG island methylator phenotype. So when people look at DNA methylation, they found that a lot of genes, the promoters become, in this case, more methylated than the adjacent normal. So this is comparing the DNA methylation levels with the normal. So these patients have a much more stronger DNA methylation patterns on its genes, gene promoters. And interestingly, when they look at the mutation patterns, they realize that all of these in colon cancers, at least most of them, has BRAF mutation. And so you can see there, sometimes this is epigenetic phenotype, but then it might be associated with a gene mutation, which is kind of interesting, right? This is another example. This is a glioblastoma in 91 patients. Again, these are coming from different studies. The rows are the CPG promoters, and the columns are the different patients. And you can see this is one group of patients that has very, very unusual DNA methylation patterns in here compared to the normal. And this, again, is called the GCIMP. So G means GBM. CIMP means, again, CPG island methylation phenotype. They look very, very different from the other patient. It turns out that the patient in here mostly have IDH1 mutations as well. So again, you can see sometimes these DNA methylation phenotypes are associated with a gene mutation. This is another case with gastric cancer. And if you look at, again, all the genes, DNA methylation compared to the adjacent normal, and then the columns are patient, and this group, again, is kind of an outlier. And turns out these are all related to EBV infection. So these are a particular type of viral infection, which is known to be related to causing cancer, because if the liver or the gut has these viral infections, the cell has to kind of repair itself all the time. There's usually some immune response, chronic immune inflammation, and they are known to induce cancer. And you can see here, all the EBV infected cancer types have a very special type of DNA methylation pattern. This is another case with kidney cancer, 161 patients looking at the methylation. You can see, wow, there's a group of patients that also are very, very different. It's a renal, renal is kidney, CIMP phenotype. And turns out they are related to a metabolism mutation, which is also quite interesting, right? So sorry, this is out of order in here. And so in these two types, interestingly, they are also associated with microsatellite instability. And so there's, it's related to DNA instability. And potentially interesting is that, do you guys remember from the immunotherapy talk? The immunotherapy, so if the DNA has a lot of mutation, like heavy mutation load, and also especially if they have macrosatellite instability, this now become a biomarker. Patients with MSI should then be treated with immunotherapy, PD-1, and they usually respond very, very well, right? So you can see here, epigenetics is linked to genetics and also now linked to DNA instability and linked to immunotherapy response, right? And these two types are related to TCA cycle, which is metabolism. Sometimes the DNA methylation patterns reflect a particular metabolism differences in a cancer. It's known that in general, cancers have very, very different metabolism from normal cells. And especially if you have mutations on some of these genes, it can really change the metabolism, which can also be reflected in the DNA methylation patterns. Okay, so these are, and interestingly, for the patient with these mutations, now when people are looking at their survival, so every time you look at this, oh, what happens with this subtype? You ask, do you see differences in their clinical outcome? So we know, for example, with the CIMP phenotype or MSI status, you could use immunotherapy, right? But then for these two types, we can ask, do they correspond to difference in response or outcome? And if we look at the survival for this particular cluster, it's great. So with any patient with IDH1 mutation, these are TCGA patients, but they are not treated with immunotherapy. But still, even for standard drug treatment, chemotherapy, or other targeted therapy, the patients seem to do better overall in their survival. Whereas in this case, those poor patients with this another metabolism differences, you can see there, they usually have very, very poor survival. So it would suggest that these mutations or epigenetic changes or the metabolism changes really will influence the patient's clinical response to other drugs. And so you can imagine DNA methylation as becoming a very important angle in looking at cancers in general. Questions on DNA methylation, at least?